Nice. All right, why don't I just start it up? Hi, everybody, and welcome back to our IVM seminar. And today we are going to go back to the roots of biochemistry, molecular EPR, with our presenter, Nina Kubatova, who's going to tell us all about gpcrs and specifically the β1-adrenergic receptor and how we can tease out equilibrium studies and further information of how the gpcr is behaving inside the membrane with our favorite techniques, EPR spectroscopy, specifically the DO experiment. So Nina, if you want to, would you take it from here?
Yeah, thanks, Thomas. So I'm coming from the Solution NMR group, and I first of all want to thank Thomas for introducing me for the EPR field and for the grateful collaboration, which I'm going to present. GPCRs are one of the largest membrane protein families in eukaryotes. They regulate signaling systems like taste, smell, vision, and many, many others. The importance of studying GPCRs was recognized by a Nobel Committee in 2012, so that Robert Lefkowitz and Brian Kobilka received the Nobel Prize. So all GPCRs can be classified based on their sequence homology. There are basically 4 different classes, and the class A rhodopsin family is one of the biggest. It makes up to 58% of all of the GPCRs. All GPCRs show a common mode of signaling transduction. They consist of 7 transmembrane helices connected by their loops in the extracellular part and in the cytoplasmic part. The diversity of all GPCRs is made up of the different loops. So basically the transmembrane domains are the same, but the loops are very different. So GPCRs are signaling proteins. The first messenger is the ligand, which is coming from the outside the membrane, from the extracellular side, binds to the protein, and this leads to the conformational change of the protein, making it from the inactive state to the active. Once it is active, it is able to bind either G protein from the cellular cytoplasmic side or arrestin. If it binds to G protein, then the signaling goes on. If it binds to arrestin, then the signal transmission is terminated. So I'm particularly interested on the oligomeric state of the GPCR in each particular step of the cycle. So for now, only one class of GPCRs, namely class C, is known to be the dimer. All other GPCRs are under debate. So there are experiments showing in vivo that GPCRs are dimer, but also other experiments compromising this and saying it's monomeric. So what I want to know is, at first, what is the conformation in the inactive state? And then I'll continue with the other conformations. So since this is the membrane protein, one of the important components of the membrane is cholesterol. And I'm also interested in how cholesterol can affect the function, conformation, structure, anything of GPCRs. Unfortunately, cholesterol itself is not soluble. So we have to work with its soluble analog called CHS. And we also choose another soluble analog, which is sodium cholate. So the difference between cholesterol and CHS is not big. It has the same hydrophobic steroid core, which penetrates inside the membrane, but it has an extended hydrophilic chain, which makes it more soluble. In case of sodium cholate, which is a derivative of cholesterol, and it also is a bile salt, it has extended hydroxyl groups, and the whole form of the molecule is divided into 2 parts. We have the convex hydrophobic form and the concave hydrophilic. So when cholesterol penetrates inside the membrane, the rigid core, steroid core, aligns parallel to the phospholipids. And this leads to the increase of the thickness of the membrane, and it also changes the geometry, the water penetration, and so many functions. In case of sodium cholate, the molecule aligns itself parallel to the surface. So instead of going inside, it is laying on the surface. And this reduces the thickness of the membrane, and it increases the solubility, and at high concentration, it can even completely dissolve the membrane. So since I'm working in DDM, the principle of interaction between cholesterol CHS and sodium cholate with DDM micelle would be pretty much the same. So it will interact with the surface in the same manner, and changing the physical properties of the micelle pretty much the same. So the system I'm working with is β1-adrenergic receptor. This receptor is mainly expressed in our heart. It regulates strength of our heart contraction. Typically, all of the GPCRs are expressed in either insect cells or mammalian cells. However, this construct was designed in the group of Stefan Grishek in the University of Basel to be expressible in E. coli. And to do so, they attached fusion proteins on the N-terminus and C-terminus, and also introduced the cleavage site. So I'm not going to go through all the purification step. I just want to focus on one particular step, which called ALAC. It's receptor-specific alphenyl agarose affinity chromatography. So as I mentioned previously, this protein is expressed in E. coli. And E. coli lacks a lot of machinery which are present in the mammalian cells. And as a result, the protein might be folded not entirely correct. So in order to end up with a totally properly folded protein, we need to separate between properly, not properly folded. So how do we do this? This protein is able to bind ligand, specific ligand, and this ligand is attached to the resin. So we pass through the protein on this resin, and only the proper folded protein, which is able to bind to this ligand, will bind. And then we just dilute it. So by the end, we are sure that the fraction that we get are totally folded and might be activated by the further steps. So to ensure you, we can get a pure protein, which gives a nice size exclusion profile and also pretty nice pure bands on the SDSPAGE. So what do we want to know first? What oligomeric conformation does this protein observe in DDM? And it can be either monomer or dimer or higher oligomers. And to answer this, out of all possible methodology, we choose DIA EPR spectroscopy. So the principle is, as you all know, we attach a spin label to the cysteines, measure dipolar coupling, and based on this, we can get the distance distribution. So if we have two cysteines, we should get one distance. We measured that on our β1 in DDM micelle, and these are the spectrum. This is the spectrum we've got. So instead of one, we have lots of signals. So how do we assign this? In order to do so, we have first to look into the cysteine distribution all over the sequence of β1. So there are 10 cysteines, which are naturally occurring, and we have not mutated anything on this step. 4 of them are located in the extracellular side and are forming disulfide bridge. So they are blocked for their spin label. 2 of them are in the middle of the transmembrane domain, so they're also not accessible for the MTSL labeling. And the rest 4 are potentially capable of MTSL labeling. So to assign the peaks, or just to figure out from which cysteines they're coming from, we performed a single point mutation on each all of them. So based on those experiments, we see that the MTSL was labeled only to 344 cysteines and cysteine 163. So we have two cysteines which are labeled with the MTSL spin label. It has to give us only one signal, but we have three. So to understand how we could end up with three signal, we looked into the structure and performed a simulation using R1 nitroxene conformation library in Explore-NIH. So we took a crystal structure, put MTSL label on those two positions, and ended up with possible location of the spin label. For cysteine 344, there were 4 possible positions for the spin label. For cysteine 163, there were 5. These simulation data were not overlaying with our experimental one. So with a little bit of playing around, we figure out that we have to remove the positions which are highlighted in gray here in order to get the best overlay with the experimental data. And as you can see here, we pretty good succeeded in this. So what does it mean? It means that only one C44 position is on there, but there are 3 different positions of C163 labeled. And the heterogeneity arises from different orientation of the spin label. And this is important because typically you would see this is a different protein conformation that we observe over there, but indeed it is not. It is just a different position of the label itself. Another important point over there I want to mention is the position of one detergent molecule is this one. So the crystal structure already has several detergent molecules which were co-crystallized with the protein. And by doing the simulation, we saw that the position of particularly this one is very important to get the best overlay with the experimental one. This means that the detergent which is surrounding the protein also determine the position of the MTSL label itself. Okay, so now we have the assignment of those signals, and we know that they are coming from the different position, naming ABC from the C163. So let's look what we will get once we titrated with CHS. So we performed the titration series, and as you can see, we have increasing with increasing of CHS, the signal at 26 angstrom here is decreasing, and the signal at 34 angstrom is increasing. And CHS is known to be a modulator for GPCRs. There are different binding sites all over the place on the GPCRs. Here I mapped the potential binding sites for β1-adrenergic receptor that were found in crystal structure. So basically there are 4. 2 of them are located in the extracellular site. They are very far away from our spin label, so even if cholesterol is binding there, we will not be able to observe them. Binding site 2, which is here, is in the cytoplasmic site, but it also far away from our spin label, so we will not get any information if it's bind there or not. But luckily, binding site 1 is exactly where we have the spin label at C163. And lucky for us, exactly this position was found to be a cholesterol consensus motif, meaning that this site is specifically binds to the cholesterol. So at this stage, we're pretty lucky. We can be sure that CHS will bind there. How can we explain what's going on here? So as I mentioned before, CHS is very close to the position of 163. And it binds exactly at the position where the C163A is located. So when it binds, it just replaces one of the spin label and shifts it towards the position to be. And this is exactly what we observe experimentally. So the position at A is now occupied by a CHS molecule, and the spin label is shifted towards position B. We also performed global fitting, and this gives us the KD value of CHS binding to the GPCR 1.4 millimolar, again suggesting this is a specific binding. Okay, so let's move on to the interaction with the cholate. So similar as before, we have titration series with the cholate, but now in contrast to CHS, all those three signals do not change upon addition of cholate. But what does change is an additional signal at 26 angstrom. So upon addition of cholate, some new species is appearing and the signal gets rise. So to answer this, we have again looked to the crystal structure. And there is one study where they've got dimers, better to say planes of dimers with different dimer interface. So according to this study, there are two potential dimer interface. The one is over there and the opposite dimer interface too. So we again did the simulation in Explore-NIH and put the MTSL label on all of the cysteines and looking into the distance between the spin labels. So this is the spectra you're already familiar with. This is intramolecular distance within just one GPCR. And here you can see the intermolecular distance between two molecules in the dimer interface 1. So the distance between cysteine 344 and 344 for another subunit gives us a signal at around 26 angstrom. And if we sum up all of these signals coming from different distances, we get very good overlay with experimental data. Again, supporting that the dimer interface 1 is what we observe. We also check the dimer interface 2. And in this case, the distances between the spin label were so far away that no chance we could get a good overlay with the experimental data. So if this is not enough to confirm you that this is a dimer, I also have a gel. And this is SDSPAGE. Here is the first lane is beta-1 in DDM micelle, nice monomer. Then addition of cholate, there is a signal appearing over there, which corresponds to the dimer, and no band upon addition of CHS. So at this stage, we know we have a dimer upon addition of cholate, and the dimer interface is dimer interface 1. Also, this dimer interface has been shown to be present in rhodopsin. Rhodopsin is another GPCR which is located in our eye, and it's very well studied. So similar to CHS, for the cholate titration, we performed a global fitting. And with this, we got KD of 35 millimolar. And this KD corresponds to the GPCR dimerization. So now we have results supporting that we have a specific binding with CHS. We do not have a specific binding with cholate, but upon addition of cholate, we observe the dimer formation. So what we did next was combining CHS with cholate. So here you can see the spectra where we have excess of cholate and lack of CHS. And we do observe the dimer because we have excess of cholate. But we also have a small shift towards the B position, meaning that the CHS is binding, but not completely. So we increase the amount of CHS. And at this experiment, we have almost fully saturation with cholate and CHS. And as you can see here, dimer peak is almost gone and CHS is almost fully bound. So what does it tell to us? It tells that the dimer gets dissociated upon addition of CHS. And actually, with this fitting, we could also say that only one molecule of CHS is needed to dissociate the dimer. So at this point, I want to conclude everything we've done and to propose you in theory what we observe. So there are two different mechanisms how cholesterol can interact. First is the direct mechanism. Direct, we have observed with the CHS. Direct means that the ligand binds to the protein, and this leads to any changes in the conformation of the protein. The indirect mechanism implies no direct interaction with the protein, but changes of the membrane. So we also have this in case of cholesterol. It penetrates inside the membrane, it changes the thickness, it changes the physical properties of the membrane, and so on and so forth. And the same thing happens when we work with CHS. It also penetrates inside, changes their physical properties. However, in case of CHS, the direct binding is what is most important, and this is what we observe in our experiment. So in case of Cholate, we don't observe any direct binding. What we do observe is indirect mechanism. It interacts with the membrane, and this somehow facilitates the dimer formation. And what's even more important, once we add cholesterol, or better say CHS, this dimer formation get disturbed, and we end up with two monomers. Well, with this, I'm at the end of my presentation. I want to end with acknowledgement. Of course, Marius is my boss, supporting all the crazy ideas during this project. Thomas, the main guy, who helped with everything with EPR, and also he performed the global fitting. Yeah, well, everyone in my group, especially Stefan Grishak, who provided us with β1 plasmid. And thank you all for your attention.
Thank you very much, Nina. It was a great talk. I agree. I really appreciate all the information, specifically the insights on how to actually deal with membrane protein, which are truly challenging for most other biophysical techniques. Now we come to my favorite part, which is obviously the question session. Before I continue, let me ask right away, does anybody have questions? And if you do so, please just either raise your hand or just talk right into it. Feel free to interrupt as much as you want. All right, while we're waiting, I would just go ahead and ask you a quick question. You showed that oh wait, actually, hold on. There was a question. Sorry.
Yeah,
how did you, near?
It's more to you, Thomas, because you calculated KD, so maybe you can answer that.
All right. Sure. Near, how are you doing? I hope you're doing fine. How do we calculate the KD? Well, we did it through a simple. Nina, can you go to the slides, or just the CHS? Perfect. So because we know that it's like a simple binding isotherm, one could use the quadratic equation. In this case, we used both. Both come to the same conclusion. We just went later on with just a simple binding isotherm, as it is a specific binding to a binding pocket occurring. Now, when we did the fitting, however, we used a global fitting where we used the raw data. We did not use population, fitted the population. We actually fitted everything to raw data. Now, as Nina said, this is a subset of traces we're showing. So in the end, we had, I don't know, 15, 20 traces, which were globally fit, where the global parameter ended up to be the KD, and pretty much the population of what is that? 31 angstrom distance, the 34 angstrom distance. Does that help you, Neil? Yeah.
Can't really see the question.
Oh, okay. Bella is asking that in this case, we're looking at the dimerization event. And because we have a dimerization event occurring and we have labeling occurring, we have a modulation depth that normally increases with a number of paramagnetic centers present to it. But in the case presented here, we have a variety of modulation, and it doesn't always follow this trend with an increase. Even though we have dimer form, we don't always see a modulation depth increase. And
Hi.
So I guess let me just answer that. Thanks, Bella, for the question. Yes, you're right. Normally, we would see a modulation depth increase, and it normally would be a dead giveaway for the dimerization event. However, in this case scenario, for once, we're dealing again with a membrane protein now. I'm just going to say it's not a perfect labeling occurring. And so we disregarded that information for now. And we also had an additional information, as Nina showed the SDS gel, that there's truly dimerization occurring. And we probably just expect that there's some stuff happening that we can explain. Sorry. Thanks.
So,
Daniela,
yeah.
nice to see you. It's been a while.
Hi. Yeah. Hi, thanks for the talk. Yeah, I'm also a bit confused. You have a complicated system because you have both the dimerization and you have the binding, right? And your binding, if I understood correctly, changes the dimerization.
Yeah, that's correct.
So this is a complicated calculation, I think. So maybe you need to first calculate the KD of the dimer independently and then stick it into your equation somehow, I think.
Well, we have calculated just the dimer first, which was acquired just with Cholate. So once we have only Cholate, then we get only dimer. And this is like a separate mechanism. And here with the global fitting, we got the KD for the dimer.
Okay, so here you have the global fitting of the
dimerization.
Okay, so you plot the modulation depths, right? What do you plot here?
In this figure.
What is your observable? You look at relative intensities of the peaks.
Population, that's right. Population of distant distributions.
Because you have some that are inter and some that are intra, or what?
There is only one which is arising, and this one is 26 Armstrong. And this peak is coming only from the dimer. So we can pretty easily read this one from the intra molecular.
I see. Because if you had only one cysteine, then you could go for the modulation depths. And this would be probably more accurate. Okay, now I understand. Okay, thank you.
Nina, can you one more question? Can you like iterate on like when we calculate where those distances come from? I know you explained it already, but can you iterate again like that? When you modeled it, how did you come up with a CHS distance? Like how did you find out the CHS distance? Can you iterate on the program you used?
You mean CHS?
Yeah, there you go.
Well, from the crystal structure, we see that CHS is binding over there. So the distances before CHS binding, we could already assign, and CHS is binding exactly the position where one of the spin label is located. So it basically replaces it and moves it towards another position. And this is exactly what we observe during our titration series, that the biggest one, which corresponds to the position A, decreases, while B, which was smallest one here, is increasing. So these are the distances from the crystal structure simulations.
So then it's just to iterate again. So that actually makes it necessary for that detergent molecule to be present on the site of the GPCR, which makes sense as it was observed multiple times in the crystal structures. Right?
Well, the detergent molecule is not specifically related to the binding of CHS. It is generally related to the proper distribution of MTSL label. So it doesn't matter if we have CHS or we don't have CHS, we always have to take into account the position of one detergent molecule on the surface. Otherwise, we will not get the good overlay with the experimental data. So apostate, meaning with no sterol, it's also very important to have it over there.
Okay, great. That makes sense.
And for example, for the dimer, the crystal structure was without any detergent molecules. It was super pure. There was nothing extra. So I had to put it extra, this detergent in this position, in order to get the same distribution.
Okay, that makes sense. So then, but that is very fascinating for me because it really pushes the detail, like what can be observed simple just based on MTSL distribution on the chemical environment. That's very fascinating. So thanks. Please ask away. Any other question? Okay. When you presented the cycle, you said can you go over it again? You mixed CHS and Cholate together, right? And both of, if I'm correct, they both had opposite effects from each other. So that means there's like both of them can actually regulate the dimerization of GPCR in the membrane. Is it like in biochemistry? Is there like information? Is like could that apply to a biochemical regulation mechanism of GPCRs in the subsequent pathway?
So Cholate interaction is indirect, meaning what we observe here is changes in the membrane. And this is what facilitates the dimerization. While CHS is the direct binding, and this is like way more important because the specific binding moderates, like it can just impede the conformation, the active conformation. So what we know from the studies in vivo is that the dimer of beta-1 is present. It's not known in what amount, but it is present. However, we also know that once we have CHS, it will interact with beta-1 receptor as allosteric modulators. So what it does, it binds, and then the protein is not able to change to the active conformation anymore. So CHS not only inhibits the dimer formation, but it also inhibits the activation of the protein.
Ah, okay.
However, the question whether dimer is needed for the activation is still open.
Hmm. So that means it's a pretty much ongoing, like but then that's kind of important, right? To know if there's a GPCR dimer and how that subsequently will apply to it, because it turns out, as you mentioned, GPCR is a pretty big thing. So it's not. It sounds like people have been debating about it quite a while, and that seems to be.
Still do.
That's good. Yeah. Any other questions? All right, well, with that in mind, I want to thank the speaker and also everybody else for attending the seminar. And I hope to see you guys in 2 weeks' time again. And thank you very much. Thank you.
Thank you.
Bye, Daniela. How are you doing?
Good.
That's been a while. I was
Good.
talking to your grad student. She was very fun. I forgot her name.
Where? When?
She emailed me. I give a talk in Brooker. It was a Brooker seminar, and I give a talk about HIV. And she was like, "Oh, Thomas, how are you doing?" And then we had a Zoom meeting where she was just asking question of how to apply DIO to live cells for HIV drug detection and
So
drug screen.
probably Yasmin.
Oh yeah, just yeah. She was telling me about her idea book. We're like, yeah, it's good to have crazy ideas. And it was wonderful talking to her. She was really, I thought she was very nice, especially that she could apply like crazy ideas and can think about it. It was very awesome. I enjoyed it a lot. She was really
Okay.
good.
Okay, great.
Yeah.
Okay,
Anyway.
take care.
All right. Bye, Daniela.
Bye-bye. Bye.